Ajit Datar to kindly deliver his lecture. Dr. Ajit Datar, sir, please.
Yeah, thank you, ma'am. Thank you, Dr. Kavita and Dr. Nandini, both of you, for inviting me for this particular webinar. And also, I welcome again all of you who have joined this particular webinar. I think number is more than 300. That is what I've heard. And in such a large number, all of you have come. I hope I will be able to give you something from this lecture. So as the topic has been already disclosed and you already know it, that I'm going to talk on mass spectroscopy basics and applications. Well, I'm analytical chemist, and what you are seeing in front of you is my friend who is nothing but analytical chemist. As you can see, he's playing with some 5 objects, and those objects are sensitivity, selectivity, separation, structure, and speed. These are 5 important S in analytical chemistry. We always talk about this. We always talk about sensitivity. We always talk about how the method could be best selective for a particular analyte of interest. We need to carry some kind of a separation, which could be simple thing like filtration, centrifugation, then liquid-liquid extraction, solid phase extraction. And of course, the best method for separation is inverter chromatography. Then we also talk about sometimes the structure of a compound, unknown compound, and we need to find it out in many of the applications. We always do a lot of quantitative analysis, but we also need to do some qualitative analysis sometimes, and that is possible. And then all the kind of measurements which we would like to do, we would like to do it as early as possible. In fact, the punchline for chromatography is that carry out a separation in less time. So you have to get a separation in less time. So time is important in industry, time is important everywhere in life. So we have to do the separation as early as possible. We have to complete a method as early as possible. Now, while playing with this 5S, something needs to be suppressed. And as the juggler, the analytical chemist is trying to balance himself on a platform which itself has a gyratory motion, he has to control these 5S by suppressing something, and which are sampling error. Sampling error is not only the how you withdraw the sample, how your sample is representing the bulk of the material, but it is also how you prepare your sample. So sampling error involves both these aspects, the sampling, true sampling from the bulk, and also how you prepare the sample. If you commit any error there, your entire analysis will be wrong. Then you have always some kind of a background which can come because of the matrix which is present along with your analyte. So background is always there, and you have to make sure while you prepare your sample, you suppress this background. And then there is always a noise associated with any electronic measurement. So you have detector which will give a noise. There is a power supply which can give a noise. And even your sample matrix also can give you a noise. So noise is inherent part of the system, but you have to make sure that you reduce it to the lowest level. And that is why when we try to define sensitivity of any analytical tool, we always talk about signal to noise ratio. You must have heard these terms many a times, and there are 2 terms which are very popular. One is the limit of detection, other is limit of quantitation. And for limit of detection, signal to noise ratio has to be 3:1. And for limit of quantitation, it has to be minimum 10:1 or more. Okay, so we always define sensitivity of any analytical tool with respect to signal to noise ratio. We calculate LOD and LOQ on the basis of signal to noise ratio. So this juggler of mine, or what I call it itself is analytical chemist, he plays with this and try to balance himself. And if you can manage to do these errors, reduce these errors, you will be able to manage to play with 5S very well. Okay, so this is the concept of analytical chemistry, and why I'm talking to you about this when the topic is mass spectroscopy, basically because mass spectroscopy is the only technique which deals with all the 5S. It also gives you much higher sensitivity, much higher selectivity. It does by itself a separation in addition to chromatography. It gives you structure, and then all that can be possible with a high speed. So that is why this is very important to understand these concepts. Now, where mass spectrometry can be used, or what are the applications? And these are some typical applications. Probably you must be knowing some of them, and you may not be knowing some of them. to detect and identify the steroids in athletes. So athletes work under the influence of steroids sometimes, and they need to be caught. And for that, the tool which is used is nothing but mass spectrometry, either GCMS or LCMS. Determine the composition of species found in space. Okay, so when a space shuttle goes and it collects some samples from the higher space, then whenever it comes down, whatever is there has to be identified, and that is done by mass spectrometry. Determine whether honey is adulterated with corn syrup or sugar syrup. Now, when we talk about pure honey collected by honeybees, we would like to have it a real pure form, but many times it is adulterated, and this adulteration can be found out by mass spectrometry. Locate oil deposits by monitoring petroleum precursors in rock. Okay, so when any oil exploration is done, they try to find out from the place, after digging few feet, the rocks which are collected, they try to find out if there is any petroleum precursors or markers are there. And if they are there, they go deeper, and then probably there are good chances of having oil coming from there. Then monitor the fermentation process for biotechnology industry. And a lot of pharmaceutical compounds are coming through fermentation, especially penicillin itself is a fermentation process. Detect and measure dioxin content in meat and milk products. Dioxin is a very toxic compound, and it is found in milk products and meat. And that has to be measured because, as I said, it is highly carcinogenic, highly toxic, and it is very essential to be measured. And especially when we are exporting something, it has to be measured, and its level has to be below what we call as maximum residual limit of that. Determine gene damage from environmental contaminants, and there are a lot of toxic contaminants which can spoil the gene, and those can be considered as genotoxic and carcinogenic compounds. So we have to find out that, and mass spectrometer is especially used for that. Establish elemental composition of semiconductor material. It's the highest purity which is required to manufacture any semiconductor material, and any minor impurities which are inorganic impurities which are present need to be measured, and they use ICP-MS kind of instrument for that, and mass spectrometer of course is a part of that. Detect explosives used at the site of explosion. So when any explosive is used, forensic science people try to collect the debris from there and try to find out which explosive was used. Okay, so that's also is done by mass spectrometry. Detect the disease biomarkers. So a lot of work is going on in to finding out the biomarkers which are responsible for disease. This could be metabolites, this could be protein biomarkers. So they try to identify the biomarkers so that the disease state can be identified, what state the disease is, especially cancer biomarkers, and you can identify the disease at much earlier stage, and the patient can be cured much faster. Assist in the entire drug discovery process. So the entire drug discovery process, in various stages of it, you require a mass spectrometer. Find out the structure of active ingredient in phytochemicals. So when we try to extract the phytochemicals from any plant origin, we try to find out which is the ingredient in that, and that can be done by using a mass spectrometer. Well, this is a short history of LC, GC, and MS. So MS has started somewhere in 1905 along with LC by itself, and then MS grew like anything. Initially, the MS was in the hand of the scientists who were working in physics. Then it moved to the chemistry, and now today it has become a part of biology. So it is from physics to proteins it has moved. So currently it is maximum used in the analysis of proteins or identification of structure of proteins. So I'm going to talk about some of the aspects like what is mass spectrometry, what information can it provide, where are mass spectrometers used. MS has hyphenated to chromatography, especially GC and LC, because the MS is used in most of the applications with chromatography. How does mass spectrometer work? So what are the components of a mass spectrometer? What is called as MS or tandem MS? Then applications and some internal sites. I hope I have some of them, but let us see. If I don't have, I can put it across afterwards. What is mass spectrometry? It is an analytical tool which measures molecular weight of the analyte and also find out the structure of the molecule by using its fragments. It can require a very small amount of sample. Picomolar concentration of sample is sufficient. It measures mass up to fourth or fifth decimal level, so molecular weight can be measured up to that level. Mass range can be measured up to 2 million Daltons, so very large molecular weight proteins can be measured. This means it can detect amino acid substitution, post-translational modifications of proteins, so such kind of information can be easily obtained. Can measure up to femtogram level of concentration of analyte in a given matrix, especially when I talked about dioxin, they can be measured at very low level. Well, how you get a mass spectra? Ions are produced in the source and are mass analyzer in vapor form. So there are two basic requirements of mass analyzer. It wants the analyte in the ionic form or charged form, and it also wants it in a vapor form. So these two are basic requirements of a mass analyzer. Then once you have the analyte in a vapor form already charged or ionized, then they are separated according to their mass to charge ratio in the mass analyzer, and there are several mass analyzers: quadrupole, ion trap, time of flight, etc. Ions of the various M by Z, they exit the analyzer and are counted by the detector. Okay, so that is how it happens. And what information you get? Mass spectrum is the relative abundance of ions of different M by Z produced in an ion source. It is called as a chemical fingerprint. It contains molecular weight information mostly or generally, structural information mostly, because it can give you some fragments which will help you to find out structure, and it is definitely used for quantitative analysis. So it can do both qualitative as well as quantitative analysis. Let us quickly go to the parts of the or components of a mass analyzer. You require a sample inlet from where you can introduce a sample. Then sample needs to be ionized, so that is done in ionization chamber. The ions which are produced get into the mass analyzer, which will separate the ions on the basis of mass to charge ratio. They go to the detector. Detector will analyze the ions and signal will be processed by the data processor. Remember one thing, that you require a very high vacuum in this region so that the ions which are produced will be able to move from the ionization through the mass analyzer to the detector without any kind of a collision with anything else. If there is a air around there, then the ions in a vapor phase will collide with the air molecules and they will get displaced somewhere and they will not be able to go to the detector. And that is why we require a very high vacuum in the region, and the vacuum level could be 10 raised to minus 5 millimeter, and that level of vacuum is required. So you require to have a very good vacuum system for mass analyzer. Sample inlet could be many. There could be direct introduction by probe or through the membrane. So there are various methods to introduce sample. Then through chromatography equipment like GC, LC, or any kind of ion exchange chromatography, it could be supercritical fluid chromatography, etc. It could be introduced through elemental analyzer, automatic elemental analyzer, where we try to analyze carbon, hydrogen, nitrogen, oxygen, sulfur, etc. So that elemental analyzer can be coupled to mass spectrometer, and it is generally used for isotope ratio of these stable isotopes, and that is used in many applications. Then it can be coupled to ICP and can be called as ICP-MS, and that can give you very low level of elemental analysis and also can give you isotope ratios. So it is very useful tool for elemental analysis. It can be coupled to TGA and DTA also, thermogrammetric analyzer, differential thermal analyzer, to find out the volatiles which come out from the techniques, and those volatiles can be analyzed. So that's also one of the way of introducing sample into the mass analyzer. There are many other methods, but most common sample inlet systems are GC and LC, because those are most popular, GCMS and LCMS. Those are called as hyphenated techniques, and those are really very, very popular. Okay. So what is the advantage of connecting chromatography to mass spectrometry? Chromatography gives you only one dimension, that is retention time. And on the basis of retention time of the standard and of the analyte, you can say that the sample could be this or analyte could be this. So it only depends on the retention time, and there could be many compounds which can also have the same retention time as your analyte. So it is very difficult to justify the presence of a compound only on the basis of retention time. Mass spectrometer gives you additional data. dimension, it gives you molecular weight information. And if you have molecular weight information and the retention time, both of them are matching, then you have more confidence in saying that the compound could be present. And if you get the fragments of that, that is a third dimension. And because of that, what we call as a mass spectra. And if this mass spectra matches with that of a standard, the molecular weight is matching, retention time is matching, and the mass spectra is matching, you have definitely more confidence in saying that this is my compound. And that is how chromatography, when connected to mass spectrometer, gives you a lot of advantages. We have, after sample introduction, we have the methods of ionization. And there are two types of samples generally we encounter. One is the samples which are volatile or can be converted into vapors. And then there are non-volatiles, which cannot be converted into vapors very easily, or they decompose before their boiling point. So it is not possible to vaporize them. So we need to analyze them also by mass spectrometry. So there are two aspects. One is volatile samples, other is non-volatile samples. If we have volatile samples, generally we use GC. If it is non-volatile samples, we use LC. And then we have two different branches. One is GCMS and other is LCMS. But whatever it is, we have to ionize the sample, either in case of GCMS or in case of LCMS. I use 3 punchlines to define 3 techniques: GC, LC, and MS. So if you ask GC what you can do, it will tell you that if you can evaporate, I can separate. If you ask LC what you can do, LC will tell you that if you can dissolve, I can resolve. This is a line which I have taken from the book on LC by Snyder. And then for mass analyzer, if you ask it what it can do, it will tell you that if you can ionize, I can recognize. And that is what mass analyzer can do. It can give you the compound which is there in your sample. So it can recognize that compound. Now, as we can see below, there is something which I have mentioned, derivatization. Some of the non-volatile samples can be converted into volatile by some kind of a reaction, which is called as derivatization. And by derivatization, those compounds can become volatile, and then they can be analyzed by using a GC, and then of course a GCMS technique. So there are a few compounds which can be derivatized, and they can be measured by using a GCMS, even if they are non-volatile. So if we come to the ionization techniques for volatile, there are two techniques called as EI and CI. For the short of time, we'll just cover a little bit of EI. We'll not cover a CI. Okay. So electron ionization, where we use a very high energy electrons which are bombarded onto the molecule, which is 70 electron volts. It would produce M plus radicals. So one electron from the molecular orbit will be knocked off, and it becomes a radical. And as one electron is lost, it becomes a positively charged radical. Then as the energy is very high, the molecule can get fragmented. So it produces abundant fragment ions, and each fragment has its own molecular weight. So we get the molecular weights of all those fragments, which will help us in finding out the structure of that molecule. There is one drawback, that some cases, when the molecule is very fragile, we may not get a molecular ion. So M plus may not be visible in some cases. Well, there is one more advantage, whether it is a fragile molecule or whether it is a strong molecule, you always have a library searchable spectra. That means once you use 70 electron volt as a default energy, you get the spectra which is common every time, or which is same every time. So for any instrument, whether you work it on today or after 1 year, you will get the same spectra for the same molecule. Manufactured by manufacturer A or manufacturer B, you will still get the same spectra. And that is why a library can be created. And there are 2 commercial libraries available for that. And those libraries can be used for finding out the structure of your unknown compound. And if it matches with the structure available in the library, you already get some information about your compound. And there are more than 3 lakhs of compounds and their spectra which are stored in this library. This is how electron ionization works. We have a filament through which, when we pass a current through this, the electrons are generated. They go towards anode. From your sample GC inlet, the molecules are coming. They get ionized, and then they are pushed through this lensing system into mass analyzer, which is beyond on this side. And that is how the ionization is happening in case of electron ionization. Let us take some arbitrary example. ABC plus electron can give you ABC excited, not interested. Then it can give you ABC plus two electrons, one electron which is bombarded and one electron which comes out from ABC. And then it can give you a lot of fragment because ABC itself is unstable. So it gives you AB plus, BC plus, AC plus, A plus, B plus, C plus. So a simple molecule like ABC can give you such a complex spectra. And then each one of these, AB has its own molecular weight, BC has its own molecular weight, A has its own, B has its own. So from this information, you can come to a conclusion what this ABC could be. Of course, in addition to that, when you have electron ionization, you have a library available, which can also give you some same information, which you can generate from the analysis of this spectra. Let us look at one particular compound and try to understand this. This is spectra of acetophenone. And this spectra gives you molecular weight information. 120 is the molecular weight of this compound if you calculate. So this is 77, 28, and 15. If you add this, it becomes 120. Okay. Then you are getting a peak at 105, which is a highest peak and which is called as a base peak. This is called as a molecular ion peak. The peak which is highest on this M by Z axis is the molecular ion peak. Then the highest peak is called as base peak, and all others can be considered as a fragmented ions. Even this is fragmented ion, but among the fragmented ion, this is a major ion, so it is called as a base peak. Now, if you look at this, this is a molecular ion ABC plus. Then you are getting 105, which is nothing but A plus B. So AB together is 77 plus 28, CO will be 105. And if you see the difference between these two, it is 15. That means the bond is broken here, methyl group is lost, and you get AB plus, which you have already seen in that equation. So this is AB plus. You are getting 77, that is A plus. And then you are getting 43, which is BC plus. So B and C together, molecular weight is 43. So a simple spectra of acetophenone gives us some idea about what this compound could be. And if you analyze the spectra properly, you will know that this is nothing but acetophenone. So that is how the spectra can be read, it can be analyzed. And of course, when the structure becomes more complex, it becomes more difficult. And there you definitely require a lot of experience to find out the structure of any unknown if you do not have any other tool like a library. Well, there is a chemical ionization. I said I'm not going to consider it, but just I'm giving you some information that it is a soft ionization. It gives you molecular weight, because in my electron ionization, I said that when you use such a high energy, the molecules which are fragile will break down totally and you may not get a molecular weight information. And molecular weight information is necessary. And for that, you have to have something which is a soft ionization. We use this technique which is called as chemical ionization. Because it is a soft ionization, it gives you less fragments, not the fragments like which you get in EI. So it does not give you good structural information. And there are some gases which are to be used as reagent gases in the ionization chamber. So what happens? One of these gas, when it is used, the electrons bombard on this gas, ionize this gas, and this ionized gas interacts with your sample molecule to give you a protonated ion. So it is a chemical interaction of this reagent gas with your molecule, and that is why it is called as chemical ionization. So one of these gases, methane, ammonia, or isobutane, which are used, and that gives you molecular weight information. Well, we are quickly going to LCMS for the reason that the time is less. We cannot use EI and CI all the time. EI and CI are the gas phase ionization techniques. They have to happen in the vacuum system. So you require a very high vacuum where you admit electrons onto the molecule and molecule gets fragmented. Now, such a kind of a technique cannot be possible because your compound needs to be in a vapor form for electron ionization and chemical ionization. And if you have non-volatile compounds, it cannot be vaporized so easily. They will decompose before their boiling points, or some of them have very high boiling points, and generally it is not possible to vaporize them under normal conditions. So you cannot analyze these compounds by EI and CI. So you require different techniques. So involatile was found to be a real challenge for mass spectrometry. A lot of research work was carried out so that involatiles can be analyzed by mass spectrometry. There were few techniques evolved like field desorption, fast atom bombardment, secondary ion mass spectrometry, thermospray, particle beam, moving belt technology, and there are many more. I'm not going to talk about each one of them. It is impossible to cover. So I'm just giving you some thing which is possible to be covered in this. And then, as I said, involatile, a real challenge for mass spectrometry. For some time, it was felt that in volatiles, generally we use LC for analysis of non-volatiles or thermally labile compounds. So they tried to combine LC with mass spectrometry, with thermospray or secondary ion mass spectrometry or FAB. These techniques can be used along with LC, but then these were miserably failure. And they felt that LCMS may not be possible at all. So I put a cartoon saying that these two species cannot come together because one is in water like liquid media. Mass spectrometer works under vapor media, and because of that, these two species are impossible to come together. There is another reason for it, that LC mobile phase, if I use a mobile phase like water, it would generate too much amount of gas to be handled by vacuum system. In case of a GC, we use 1 ml per minute flow of a carrier gas. Here we are using 1 ml per minute flow of a liquid mobile phase. And if you consider water, 1 ml of water would generate 1.2 liters of a gas at NTP, which will be 1200 times more than the carrier gas which we are using in case of GCMS. So if you allow this much amount of gas getting into vacuum system, you will spoil the vacuum system and it will give you huge background. And because of that, LCMS was considered to be a technique which cannot be in existence. Well, people worked on that. People never left that idea of having LCMS in reality. And then they came out with some modern methods like electrospray, atmospheric pressure, chemical ionization, and then they use MALDI for protein analysis, which is not LCMS technique, but by itself, it is a technique for very large molecular weight proteins, non-volatiles. They can be analyzed by this. Well, we initially talk about matrix assisted laser desorption ionization MALDI for the reason that it is one of the popular technique for proteins which are highly non-volatile samples, or a lot of biopolymers or even industrial polymers can be analyzed by MALDI. It is a laser beam which is used. Laser is bombarded onto a matrix on which we keep the sample. So matrix which is used is benzoic acid derivatives, especially synapenic acid or cinnamic acid kind of benzoic acid derivatives on which we put the protein sample. And when we bombard laser on that, the benzoic acid derivatives get ionized, get into vapor phase. And along with that, the sample which is protein sample which is loaded on that also gets into ions, and the proton gets transferred from benzoic acid to the protein molecule. It gets ionized, gets into vapor phase, and then can be separated by mass analyzer. It can give you intense matrix ions because the matrix molecules also get ionized. Used for proteins and a lot of biopolymers, and it can analyze proteins up to 2 lakhs or more than that molecular weight, and more than 1,000 samples can be analyzed in 1 hour. That's the kind of a speed MALDI instrument can have. It is generally used as MALDI TOF as the instrument. This is the MALDI-TOF results, and you can see the one of the protein which is analyzed has a molecular weight of 66,000 Daltons. Well, we come quickly to the techniques which are used with LCMS, and they are called as atmospheric pressure ionization technique. And there are 3 of them: electrospray, atmospheric pressure chemical ionization, atmospheric pressure photoionization. Now, only difference is that all the previous techniques which were invented, which we could not study because of the shortage of time, they were working under vacuum. So the ionization was happening under vacuum. Here, the ionization is happening at atmospheric pressure. So this atmospheric pressure has made a lot of changes. And because of that, LCMS is possible. So LC can be coupled to mass spectrometry. And that is what we are going to talk about. So we'll study these 3 techniques quickly. This is called as API, atmospheric pressure ionization electrospray technique, which is invented by 2 scientists, Dr. John Fain and Dr. Mann. And this technique involves passing the analyte HPLC mobile phase flow through a capillary. Around this capillary, there is another capillary through which we pass a high pressure nitrogen gas, nebulize whatever is coming out into droplets form. So you are getting droplets here. a spray is formed, which is called as electrospray, because we are applying to this capillary a high positive voltage. So the spray which comes are having positive charges onto those droplets. Now what happens as these droplets come into this area under the atmospheric pressure, where there is a flow of nitrogen, and because of that the droplets will start evaporating. The charges sitting on the droplets will start coming closer together. Now there will be two forces acting on the drop. One is surface tension force, which will try to keep the drop intact. Another is the repulsive forces, the Coulombic repulsive forces. As the positive charges start coming closer together, these repulsive forces will act opposite to surface tension forces. And as the drop shrinks further, the repulsive forces become more stronger than surface tension forces. Drop shatters into pieces. And then finally, if the drop has molecule sitting inside, the positive charge goes onto the molecule, it becomes a positively charged. And all the solvent getting evaporated, it comes into vapor phase and then gets into mass analyzer through the opening here. And there are some kind of a vacuum uh, phases here. In the first case or first phase, we evacuate it to 10 raised to -1. In this, we evacuate the entire analyzer to -3, and as it comes to actual mass analyzer, the vacuum level is -5. So there are 3 stages of vacuum system which is applied to get the correct vacuum which is necessary in case of mass analyzer. That is how this works. So it's called as atmospheric pressure ionization system. It works for proteins, peptide, nucleic acids, lots of pharmaceutical compounds, natural products, solid polar compounds, and goes up to 150,000 Dalton molecular weight. So very high molecular weight samples can be analyzed by using electrospray ionization technique. Then these are for polar compounds. So this technique is for polar compounds. We require to analyze semi-polar compounds, which are also thermally labile. And for that, we have another technique. Okay, this is the picture of 3 scientists who developed these techniques. ESI was developed by Dr. John Fenn and Dr. Mann, and MALDI was developed by Koichi Tanaka, who is working with Shimadzu. And these 3 fellows got the Nobel Prize in 2002, and they are sitting on the front bench in the function where they were awarded a Nobel Prize. Well, this is the summary of electrospray. It is atmospheric pressure ion source. Smaller molecules may be singly charged. High molecular weight samples like proteins may get multiply charged. And molecular weights of up to 150,000 Daltons can be measured accurately. We go to the next one, which is called as atmospheric pressure chemical ionization, which is used for semi-polar compounds. It is slightly different. It also, of course, happens in atmospheric pressure, where these molecules which are coming through your mobile phase are heated so that your solvent and the molecules both get evaporated. So it is vaporized. So everything is in gas phase. There is a corona discharge needle. So when you apply a very high potential on the small area of a needle at the tip of the pin of the needle, what will happen? The voltage cannot sustain there. It will fuse and it will form a corona, it will discharge, and there will be high temperature here. This high temperature will ionize the solvent molecules, and these solvent molecules will interact with the molecule of our interest to ionize it. So it is a chemical ionization. You ionize the solvent molecules, and then in turn you ionize your sample molecule. So that is why it is called as a chemical ionization technique. It is used for semi-polar molecules. So APCI ionizes a wide range of compounds, works better than electrospray for non-polar compared to electrospray non-polar compounds or semi-polar compounds such as lipids and steroids. This disadvantage is that you require high flow rates compared to electrospray because you require solvent vapors to be there for getting ionized. Compound must be thermally stable because you're heating the compound to something like 400 degrees centigrade to vaporize it. So your compound has to be stable. And of course, for any LCMS, you require volatile buffers. You cannot use a non-volatile buffer like sodium phosphate, etc. Okay. We go to the next ionization technique, which is called as atmospheric pressure photoionization, which is used for non-polar samples. And this is photoionization. So you have a ultraviolet lamp, which is giving you ultraviolet radiation. Your sample is coming nebulized and in form of spray, it is coming here. The sample molecule gets ionized, gets vaporized, and then enters into the mass analyzer. And that is how the whole process happens. Now, if the sample gets ionized by itself, by using the photons, it is well and good. But if it doesn't, then you have another method which is called as dopant APPI, where some molecule like toluene gets ionized first to form a dopant ion, and this dopant ion interacts with molecule to give you a molecular ion, and that will get into the mass analyzer. So there are two techniques, direct APPI and dopant APPI. It can be used for some molecules like fluorothene, hydrocortisone, estrol, etc. So we will look at the techniques like EI, which is able to do a lot of non-polar samples, which are volatile samples. We have some FAB technique which we did not study, but it can go up to 5,000 Daltons and do a lot of polar samples like peptides and sugars. Then we have TSP, which we also did not study, but which can do up to semipolar samples up to something like 5,000, 2,000 Daltons roughly. APCI can do semipolar samples up to 1,000 molecular weight. Then we have APPI, which does non-polar samples up to 500 molecular weight. And then we have ESI, which can do 150,000 Daltons with large number of polar and semi-polar samples. So if you consider these three techniques, ESI, APCI, APPI, all the three techniques together can analyze highly polar, semi-polar, and non-polar samples. And because of these three techniques, which are called as atmospheric pressure ionization techniques, LCMS is possible. So the marriage between LCMS is successful. I always call hyphenated techniques as marriage of convenience. So the marriage between LCMS is possible. And I put another cartoon to indicate you that the cock in the previous picture has compromised. He said, I cannot marry fish, but I can marry duck, which can also exist in water as well as in atmosphere. And the atmosphere has made the change. Marriage is successful. You can see a chickling, which doesn't say quack quack, doesn't say chick chick, it says quack chick. Okay, so that's about LCMS. And this is how the difference is now. Now the ionization is outside the vacuum. It is in the atmosphere. And after that, we get into the mass analyzer. So whether you have a GCMS or LCMS, ionization has happened, ions have entered into mass analyzer, and then they will get into the detector. So we have to talk about mass analyzers now quickly, and the time is running. And I know I started a little late, so I can request madam to give me another 15 minutes and I will be able to complete. So these are the different mass analyzer. Again, because of the shortage, I may not be able to cover all of them. I will try to talk on two main ones, which are called as quadrupole mass analyzer and time of flight mass analyzer. So quadrupole mass analyzer is very simple. It has 4 rods, and these 4 rods are very compact, about 25 centimeter to 30 centimeter in length, a foot size, roughly a foot scale which we have, that size. And the diameter is about 8 millimeter, okay, so less than a finger. So they are very compact, and they can be held in hand like this, and then we can apply a DC and RF voltage on this. As I said, the ions can be moved by applying either electric field or magnetic field. So we are applying here the both direct current as well as radio frequency voltage to these ions in such a way that the one ion at one DCRF voltage, a particular DCRF voltage, gets stabilized and go to the detector. At that voltage, all other ions, I have only shown 2 ions, but there could be many ions, all other ions will get out of this trajectory, or they will get discharged onto the rod and they will not be able to go to the detector. At some other DCRF voltage, maybe the ion which is shown by this red or orange color will go to the detector. At that time, this green ion will go out somewhere. So that is how, if I scan this DCRF voltage, I am able to detect all the ions one by one. They will get their own chance to go to the detector one by one, and I will be able to measure all the ions by scanning DCRF voltage. So I can apply this quadrupole in a scan mode where I can see all the ions from low mass to high mass as I scan this DCRF voltage. I can get a mass spectra of all the ions by using this phenomena of scanning DCRF voltage. This is one way. Now, if I want to measure continuously this green ion, because I know this ion, I know this is present in my sample and I want to quantitate it. Now for quantitation, I want it to get detected maximum number of times. If I scan it, it will get detected only once, and then all other time it will be going out. But then I can do one thing. I can keep this DCRF voltage fixed so that all the time this green ion will go to the detector. So I can measure its concentration much better now, because all the time this green ion is able to get into the detector. I am able to count it more number of times. I'll be able to detect it at very low level. So when I monitor this mass analyzer in this mode, that means I'm only measuring one single ion of my interest. Then it is called as I'm operating this quadrupole in single ion monitoring mode. So there are two modes of operating this quadrupole. One is in scan mode, other is in SIM mode, that is single ion monitoring mode. Scan mode is used for qualitative analysis. SIM mode is used for quantitative analysis. We go quickly to the time of flight mass analyzer, where we have a plate to which we apply a very high positive field. The ions are coming from one end in front of the plate, and when we apply a pulse of positive charge onto the plate, and if all these ions are positive ions, they will get reflected and they will start moving towards the detector. There is no other control in between. So once the pulse of positive voltage is applied, whatever ions are in front of the plate, they will start moving and they will go to the detector. But then movement of these will depend upon their size to charge ratio. So if the ion has a low size or low weight, it will move fast, because kinetic energy is equal to 1 half mv square. So if m is small, v will be large, so its velocity is large. So the small ion will move faster, larger ions will move slow, and that is how they get separated and get to the detector. So we measure the time taken by the ions as they move in this flight tube, and the time is measured in microseconds, and then we calibrate the mass analyzer on the basis of known masses and get the measurement done, and that is how we measure the mass spectra, and that is why it is called as time of flight, because we measure the time, and this tube is called as flight tube. So time of flight mass analyzer, the time which is measured for the ions in the flight tube, and this flight tube is about 50 centimeter in length, slightly longer, up to about 1 meter in length. So the flight tube is little longer and the ions move from one end to the other. Now, many times it is not used in this fashion. It is used in a reflectron fashion. That means ions are generated here. They go like that, get reverted because of the high voltage which is applied here. And the high voltage is applied in such a way that as you go deeper, the amount of voltage which is applied will start increasing. So if ions come like that, they will start coming out like this. Is there any problem with communication? No. Okay. So let us go back to this slide. Now, when ions come in front of the plate, they are in vapor phase. So they will never be at the same distance from the plate. They will not stand here uniformly from the plate, uniform distance from the plate. So some ion may be here, some ion may be here, some may be here. So like that. So what will happen when we apply positive charge to this plate? Though ions have same mass, they will have a play, and some of them will move fast. Those were closer to the plate. Those were little away from the plate will not get pushed that much. So they will start moving slowly. So this kind of a discrepancy has to be resolved. And for that, we use this reflectron. So the ions which are moving fast, suppose all the ions have exactly same mass, and just because of their position in front of the plate was different, some of them are moving fast, some of them are moving slow. So some of them are moving fast, they will go deeper inside and turn. Some of them which are moving slow will not be able to go deeper into this field, which is applying opposite, and they will turn here. So what will happen? Both of them will catch together again, and then all the ions of the same mass will reach the detector at the same time, and that is how they are controlled. And today's time of flight mostly is with reflectron arrangement. So time of flight mostly comes along with the reflectron every time. So this is the difference. If time of flight mass analyzer is used without reflectron, single flight tube, you get the distortion like that. The peak is not sharp. All of them have same mass, but you are seeing it differently. Here, you can see them, they are coming as a single peak because I have used a reflectron. Well, time of flight and quadrupole, there is one major difference between the two. One is a high resolution mass analyzer. Time of flight is a high resolution mass analyzer. What does it mean? Now I am showing you a nominal mass and I am showing you exact mass. These are average mass where isotopes are considered. Okay, so we are not interested in this because mass analyzer doesn't look at these masses. It looks at either these masses or these masses. If it is a quadruple mass analyzer, it will look at these masses. So we can calculate the molecular weight on the basis of this, which will be an integer. When we look at these masses, the molecular weight will come in decimals, and time of flight can measure molecular weights in decimals. So it is a high resolution machine, while quadrupole is a low resolution machine. So that's an advantage of using a high resolution machine. It is of course costly because it can give you a small differences. Now, if you look at the masses of carbon monoxide, here it will be same as 28, 12 plus 16. If you look at C2H4, that is 22 into 2, 24 plus 4 hydrogen means 4. So it will be also 28. If you look at nitrogen 2, that is diazo compound, it will also be 28 because 2 nitrogens is 28. But when you look at these masses, this will be not, carbon monoxide is not 28, but 27 point something. Then C2H4 is not 28, but it is 28 point something. So like that, that difference, a high resolution machine can find out, and that is why it can give you much better information. So time of flight also is a equally good mass analyzer. Well, we get into the tandem mass analyzer quickly. Why it is required in case of LCMS mostly. In case of LCMS, the ionization techniques like electrospray, atmospheric pressure, chemical ionization, atmospheric pressure, photoionization, all are soft ionization techniques. So they give you only molecular weight and mostly proton added. So this is a salbutamol molecule. Molecular weight is 239. I get a peak at 240 because proton is added. So I'm getting only 240. I don't get anything here. So there are no fragments. So if I do not know what is this molecule, I cannot find out the structure of this molecule. So I need to fragment it. I need to break it so that I will get fragmented ions and I will get the structure. But unfortunately, the ionization techniques which I'm using in case of LCMS are not the one which give me fragments. We have seen in case of GCMS, we have the electron ionization where we use 70 electron volts energy, and where we have shown you the spectra of acetophenone where we could get the 120, 105, 77, 43 and all those masses. Here we cannot do that. So we require something else to have the fragmentation, and there comes the MSMS. So here we are using MSMS, and what we are doing here, we are using 3 quadrupoles. So there is one quadrupole on this side, and there will be another quadrupole on this side. And this is called as a collision-induced dissociation cell. Now in the first quadrupole, we select the ion, which comes here, and this ion, this is already ion, which comes into the collision-induced dissociation cell. I admit a gas like argon in this as a leak, and then give some energy to this ion so that it starts vibrating in the field here. So it starts moving in the field, and then it collides with the argon, and because of the collision, it gets fragmented, or it induces dissociation, it dissociates fragments. And then the fragments of this are pushed into the next quadrupole, which will scan them and will give me the spectra of the fragmented ions. And then I can find out the structure of this original ion, which is nothing but a molecular ion. It is not the fragmentation which has happened here. So in the collision-induced dissociation cell, I am doing the fragmentation. So let us assume this is ion which has got a mass of 420. Now this 420 ion comes here, I give an energy to it, it starts vibrating here, and it collides with argon gas, gives the fragments, and those fragments get into the next quadrupole, which will analyze it and will give me the structure of this compound. So it will reveal the information about the structure of this compound. Okay, so that is how it works. So what I'm doing, 420 को मैंने CID cell में लिया. This is collision induced dissociation. It is called as CID cell. So 420 को CID cell में लिया, उसको punches दिया. So it breaks down, and after breaking down, it comes to the next quadrupole, reveals the information, what crime it has done, and we get the structural information. And that is what this CID cell here does. It is called as collision-induced dissociation cell, but the functioning of this is exactly like a CID cell. And that is what we require and that is what is called as MSMS. So we require a triple quadrupole like this. So we have one quadrupole here. This is a CID cell. This is the next quadrupole called as Q3. Ionization is happening here. Ions come. They are passed through various lensing system, come to the first quadrupole, where I select the ion, gets fragmented, goes to the next quadrupole, the fragmented ions, and they are measured here. Let us see how it happens. So I have many ions coming. I use a SIM mode, so only I select the ion of my interest. It goes to the next collision cell or collision induced dissociation cell, gets fragmented. All those fragments get into the third quadrupole, and that is how I measure all those ions, which will reveal the structure information for me. Fine. So I can use it in another mode. I can only select one of this ion and measure it in a SIM mode. So here I again apply SIM mode. So I measure the fragmentation of this to give me one particular product ion, and I measure this transition, which is called as MRM transition. It is too difficult to explain it in short time, but this is a good quantitative measurement where I remove all the interferences in this particular transition. Well, I can use it in a tandem, which is called as Q-TOF. So I have one quadrupole here, followed by a collision cell, and I have this time of flight with reflectron. So I can get a particular ion selected here. Without breaking, I allow it to go to this. I get a high resolution mass spectra. So I know the accurate molecular weight in 4 decimal level here. Then I take the same ion, take it to the collision cell, fragment, and all the fragments will go to the time of flight mass analyzer, and all the fragmented ions with accurate mass, I will get that, and this also will give me a very accurate information about the structure of the compound. Well, this is how the time of flight mass analyzer, so you can see the ions are generated here, they move like that, and this is how the instrument looks like. Well, we come quickly to the applications, and I'm coming closer to my end of the talk. Okay, so in pharmaceutical industry, it is used for drug discovery. I mentioned about it. So in various stages of development of a drug, right from synthesis till you apply to, or you do some clinical trials, initially in vitro, then in vivo, and then on animals, and then on human beings. So various stages of that. In all this drug discovery process, you use a mass spectrometer for finding out the structures. There is one part which is called as in drug discovery process, which is called as ADME. Okay, so metabolism is very important. So you form drug metabolites, and the metabolites could be toxic. So you have to find out structure of those metabolites. So it is again used there. Then it can be used for toxicology. So that is one of the application to find out toxins. Then pharmacokinetic studies, how the drug is absorbed in the body, how much is, we call it as pharmacokinetics, preclinical and clinical studies we do, BAB studies we do. We do formulation studies, especially degradation products after the formulation is made. So we look at them. Then we look at QC and production, identification of impurities, unexpected peaks, which you sometimes get when you try to do quality control of a particular drug. So you have to identify what those impurities are. Then contract research organizations do it for preclinical and clinical trials. I already talked about it. Generic drug companies use it for QC and production. And now there are new concepts which are come and which are very important. We talk about extractable and leachables. We talk about genotoxic impurities in pharmaceutical products, where these are very, very important. In all the products, we have to find out some genotoxic impurities like nitrosamines, etc. We have to find out leachables and extractables from the packaging. The things can get into the pharmaceutical products, into the formulation, and can give a harm. Those could be harmful to the patients which use those products. Then LCMS is used in biotechnology, protein characterization, in proteomics, metabolomics, lipidomics, all those aspects of omics. It is used. It can be used for finding out post-translational modification like glycosylation, phosphorylation, etc. It is used for finding out sequence and impurities in for quality control. You are getting now new drugs which are called as biopharmaceuticals, where you have to use them mass spectrometry for quality control. It is used for nucleotides and carbohydrates analysis. Proteins these are the, I mean, you know what are proteins. I assume you are knowing, so I'm not going to spend time on that. But these are the processes which are used for protein analysis. 2D gel electrophoresis, you use capillary liquid chromatography, use mass spectrometry, and then you have to use bioinformatics tool for identification of the structure of the protein. You have two approaches. You have a complex protein sample, you do 2D gel electrophoresis, try to find out spot which is different, then do the digestion of that spot. You can do multi-TOF on that, and then feed the data to database search. Another side, you can do a digestion directly, use convert them into peptides, do peptide separation by using LC-MSMS, and again feed the data to database search. Now bioinformatics tools will be able to find out the structure of protein which you have. Okay, so these are the approaches for a complex biotechnology or proteomics applications. It is also used in agrochemical industry, compound discovery, metabolism, toxicology, pharmacokinetics, QC and production, as it is used in pharmaceutical. It is used in organometallics to find out structure of organometallic compound. There are a lot of people who work in this area in research. Then it is used for detergents, QC, and doing competitor products, because a lot of detergents are coming with enzymes. You have to find out which enzyme is being used in the market by the competition and try to do a reverse engineering. You can use it for polymers, environmental field, water, identification and quantification of pollutants in water, especially pesticides and antibiotics in